Thank you very much for the invitation. My laboratory using Drosophila to model this disease. As you all know, KCNT1 is a gain-of-function potassium channel. So the interesting question here is how can gain-of-function potassium channel induce a seizure and a behavior defect? So that's what we're hoping to get some answer using Drosophila. So the obviously hypothesis will be if you have a more potassium current in inhibitory neurons, interneurons, now you should be able to unbalance the neural circuitry, thereby you have a hyperexcitability. So therefore, we express these channels in Drosophila inhibitory neurons, GABAergic neurons. Indeed, first of all, we verify this gain of function by express this in the muscles. As you can see here, this all have an increased hyperpolarized membrane potential, reduced input resistance, and so on, which is consistent with what we know from expression work from Lancaster, Merrick, and others. And this is a gain of function mutation. But now, if express this channel in GABAergic neurons, now you can see flies have a seizure, which is basically fly. We call band sensitive test. You basically shake the flies, not flying for the back, and have a seizure for a short moment. So this is obviously exciting, but we still don't know whether that's because it's this inhibition or not. Okay, so then the question is, what if we express this in motor neurons? In Drosophila, the motor neuron is a glutamatergic neuron, and now when we express this KCNT1 in motor neurons, we were surprised we see the similar things. In other words, in excitatory neurons, when you express gain of function mutation, you're also going to see a seizure as well. So there's a paradox here. How can you increase potassium current lead to hyperexcitability? So that's kind of question we all want to know. So, and here's what I demonstrate to you. When we actually record from the neuromuscular junction, the invoked neurotransmission after normal. So in other words, this neuron, this fly larvae has coordinate divided, but nonetheless, they're still alive. They have neurotransmissions normal as well. But when you cut axon from cell body, not just purely just nerve terminal and axon, and when we record from the synapse, as you all know, you're going to normally see this spontaneous miniature potential, which Bernard Katz observed a long time ago, which basically the spontaneous release. So this is very typical to what happened in a wild type and the controlled human KCNT1 conditions. You can see a specific miniature potential and frequency. However, when we overexpress this in motor neurons, two of these mutations, you can see this amazing large event. Okay, this is not a miniature potential. This is what we consider is a subthreshold action potential induced release. So therefore, this dramatic change, what we know about excitability in the nerve terminal. Most interestingly, this actually resistance, partially resistant to TTX. If you block voltage-gated sodium channels, you're still going to see this large event. However, if you block calcium channels, eventually everything comes down. So suggesting there's some increase in sodium channel as well, perhaps calcium current as well in this terminal, which we have not done calcium imaging to verify that. So how can that be possible, right? You have a nerve terminal without cell body, you have spontaneous depolarization, local depolarization going on, triggered neurotransmitter release. So then one way to think about this is if you increase your KCNT1 potassium channel, something has to give to maintain the excitability. So therefore, what we think, hypothesize is maybe you reduce endogenous potassium channel from flies. This is indeed what we observe. Shown here in immunosensor chemistry, in a nerve terminal, which is a neuromuscular junction. In the wild type, you have the nerve terminal membrane stained by HRP antibody. Then you have a shaker antibody stained potassium channel here. In one of these mutant fly, you can observe tremendous reduction in the average signal of shaker channel in a nerve terminal. And then we also have antibody for another potassium channel called EAG. This one, unfortunately, cannot stain the nerve terminal, but by Western blot, you can see also reduction as well. So therefore, what we think is what's happening here is you have a fly which has an endogenous potassium channel, EEG, and a shaker. And if you have you do have KCNT1 Drosophila as well, now you maintain certain neuronal excitability. But now if you increase in the KCNT1 channel in GABAergic neurons, we don't know whether this channel change or not. But nonetheless, if you silence inhibitory neurons, you're going to have a seizure as well. But on the other side of the story, if you actually increase the excitability, decrease excitability by mutant KCNT1 gain of function, now the neurons are going to try really hard to maintain that excitability. So therefore, they're going to either increase voltage-gated sodium channel, calcium channel, as well as we can detect here the decrease of endogenous potassium channel, in this case, shaker channel, as well as EEG channel. So therefore, you can see the summary here, but the key point is summarized in this cartoon version. We believe, based on the fly model, this is how the flies actually have a behavior and a neurophysiological defect in challenge of the gain of function of KCNT1 channel in this case. So what we're doing now is actually we think our flies we can use this fly to do unbiased forward genetic and compound screen, which may offer us to identify the biochemical pathways will lead to the change of this channel expression, and also genetically speaking as well. So thank you very much.
Dr. Bing Zhang, I was really interested in your research showing that there seems to be a signature in the excitatory neurons as well. Because I sort of, I thought I understood KCNT1 and I thought it was primarily an interneuron problem. So yeah, so that is really interesting. I guess what do you think that means in terms of how the disease is working?
So for the interest of time, I didn't cover everything. So I want to disclose when you express this in excitatory neurons, the fly actually have severe behavior problems in terms of larvae, they cannot crawl well. However, that behavior defect is not because they have a hyper-excitability, but rather there's a developmental defect in a sense. Normally, when a larvae crawl, they have this synchronous contraction going forward, backwards. But in this case, the two hemispheres, if you want to think about the spinal cord in the equivalent spinal cord, they actually are out of sync. So the neurons not fire together. As a consequence, the larva is going around, basically just struggling with their locomotion. So, but if you record from the synapse, neurotransmission is fine. This neuron too small cannot record from we could try really hard to record from motor neuron itself. Presumably the action potential is normal as well. Yeah.
I wonder if I could address that question too, because in our mouse model, the excitatory neurons are much more excitable and the inhibitory neurons are less excitable. And that's because a potassium channel can do two different things. If it only activates during an action potential, it will speed repolarization and lower sodium channel inactivation. That will allow neurons to fire at faster rates. And that's what happens in iPS-derived neurons where they have the KCNT1 mutations. They become more excitable. If the channel activates at more negative potentials, it makes it more difficult to depolarize the cell, and that inhibits firing. And that's what happens in the inhibitory neurons in the R455H heterozygous mouse mutation. So it's a double whammy because there are different splice isoforms of SLAC that activate at different membrane potentials and have different effects on excitability. But there's no question excitatory neurons are very much affected by this in the wrong direction that you'd want. Yeah.
Yeah, thanks for that. That's really interesting.